Everyone, welcome. I'm Jack Van Horn from the University of Virginia. It is my distinct pleasure to welcome you to this edition of our 2024-2025 Foundations of Biomedical Data Science Seminar Series. Our unique lecture series is made possible through the generous support of the Integrated Translational Health Institute of Virginia, the University of Virginia's College of Arts and Sciences, the School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since its founding in 2016, the Foundations of Biomedical Data Science Seminar Series has been at the forefront of exploring the transformative impact of data science methodologies, artificial intelligence, deep learning, and statistical modeling across the diverse themes that exist in biomedical and the health sciences. Our selected theme for this year is focused on quantitative approaches in spatial multi-omics for guiding personalized medicine, where each Friday we enjoy presentations from a wide range of leading thinkers about the issues, the promise, opportunities, and the hurdles associated with advanced genomic, transcriptomic, metabolomic, and other omic methods, along with a wide variety of data analytic opportunities presented in deep learning, AI, and spatial modeling. We are particularly pleased to announce that our 2024-2025 application cycle is open for our Biomedical Data Science Innovation Lab. And we hope that you or any of your junior faculty colleagues working in the various areas of omics-related biology as well as data science might be interested in applying. Aligned with this year's theme, we are eager to receive applications from folks working at all manner of biology, data analytics, disease modeling, epidemiology, et cetera, that involves the use of spatial multi-omics. Participants selected for our Biomedical Data Science Seminar program will utilize the content from lectures such as the one we're about to hear today as essential material in their culminating in-person manuscript and grant project development workshop to be held at the Hutton Hotel in downtown Nashville, Tennessee in June of 2025. We're excited by this year's program, and we really hope that you'll consider applying. For today's seminar, I am absolutely delighted to welcome our speaker, Professor Julianne Carstens from the University of Alabama at Birmingham. She earned her PhD from Baylor College of Medicine in Houston, Texas, in the laboratory of David Spencer in the Department of Pathology and Immunology, where she studied the cellular and molecular biology of prostate cancer, focusing on tissue crosstalk between cancer cells and their microenvironment. Following defending her dissertation, she trained at MD Anderson in Houston, where she completed her postdoctoral fellowship, and her work investigated the specific roles of cancer and microenvironmental cell types, working basically their interaction in the progression and metastasis of pancreatic cancer using genetic mouse models. Her laboratory at UAB continues this previous work in metastatic pancreatic cancer with the ultimate goal of understanding the systemic tissue and cellular and molecular events which drive pancreatic cancer metastasis and resistance to a broad range of therapies. In the spirit of our 2024-2025 Biomedical Data Science Seminar Series and theme, Professor Carstens' lecture today is entitled The Spatial and Functional Heterogeneity of PDAC Tumor Subpopulations. Pancreatic cancer is therapeutically resistant and metastatic, and Dr. Carstens' team have begun unraveling the complex functions and interactions of these subpopulations. And our talk today will discuss the findings of the tumor regulatory properties of the microenvironmental subpopulations and their function location linkage within the tumor and metastatic sites. As always, we are streaming this lecture live and for recording via YouTube. So if you're watching us on YouTube, thank you so much for joining us. Also, our specially selected 2024-2025 Biomedical Data Science Innovation Lab participants, as well as alumni, are strongly encouraged to submit any questions they have via the chat feature in their YouTube sessions. I'll be synthesizing these questions and asking them on your behalf during the last 10 minutes or so of the lecture. And with that, thank you so much, Julie. We are delighted to have you. We're really looking forward to your presentation.
All right. Thank you so much, Jack. And thank you, everybody, for your attention. So we'll just jump right into it after that amazing introduction. So today I'm really applies the use of using spatial multiplexing in omics. A lot of the understanding and the belief system of how our lab approaches spatial questions has actually been recently written up in a Nature Reviews Methods Primers, where we go over how we use spatial biology and how we apply those tools across all levels of biological questioning, from cellular to cellular and tissue and even full organism. Today, I'm also going to go over how my lab and my collaborators have used specifically multi-fluorescence imaging, spatial transcriptomics, a little bit of mass spec imaging, and some future directions here that we're going on with PET CT and MRI, and how we apply these from a biologist point of view, which is what my training is, into investigating cancer biology. So my lab primarily focuses on pancreatic ductal adenocarcinoma, also known as PDAC. It is the most common of the malignancies arising from the pancreas, and it is also sadly the most deadly, has now risen to be the fourth most common cause of death in the United States. The combined 5-year survival rate is hovering around 12 to 13%. And as you can see from where we started, when I started working on pancreatic cancer back in 2013, the field in general, the overall survival rate was in the single digits. We have been able to successfully go up now to the double digits. Pretty much all of that advance has occurred in the localized tumors where patients are eligible to receive their tumor resection. However, majority of the patients, around 50%, walk in the door with metastatic disease. And of the patients that are even eligible for resection, which compensates about 20% of the patients, 80% of those will recur with metastatic disease. And so the vast majority of the patients have untreatable, therapeutic resistant metastatic disease. So we really need to understand what is going on in the pancreatic cancer when it's no longer in the pancreas, when it has spread to other organs. So what are the common sites of metastasis? The pancreas cancers primarily metastasize to the liver. That compensates about 80% of the patients, and then followed up by both peritoneal and lung metastases. And that distinction will become important later on. So how is it that we study the cancer microenvironment and its tumor heterogeneity? I am a mouse modeler by training. We use a lot of genetically engineered mouse models. And what is nice is that we can then specifically label, ablate, or genetically modify specific cell populations and how those cell populations then interact with the tumor progression over time. We, of course, collaborate with clinical colleagues and get tumor specimens directly from the patients, whether it's from mouse or patient origin. Those tumor specimens are then analyzed with histopathology, of course, bulk tumor analysis, which is much more common and easier to do, as well as isolating out the tumor cells, doing single cell work. We do a lot of omics, a lot of antibody analysis, and a lot of culture. I've paused to show these symbols because as I go through a lot of the data that my collaborators are generated, I have these symbols showing up so you know what all these bar charts are, where they're derived from. So going back again to specifically pancreatic cancer, it is one of the most heterogeneous microenvironment solid tumors that are out there. The desmoplastic reaction, or also known as all the other cells in the tumor that are not malignant, can comprise up to 90% of the tumor volume. And then, of course, the cancer cells themselves are also heterogeneous, and they're comprised of different subpopulations. So one of the first things when I started my postdoc, working with Raghu Kalluri, was really trying to understand the fibroblast, right? So this dense desmoplasia is comprised of a lot of collagen, and the cells that primarily put down that collagen are activated fibroblasts. You'll also see in the literature being called activated stellate cells. And one of the major proteins associated with that cell population is smooth muscle alpha actin. And so a genetically engineered mouse model that has linked the promoter of smooth muscle alpha actin to a viral thymidine kinase. What that does when you add the drug, the antiviral drug ganciclovir, the cells that are expressing thymidine kinase, which in this case would be the alpha SMA cells, undergo chain termination and therefore stop proliferating. So it's a way to specifically target proliferating cells. And this model is used a couple of times, but just to pause to show that. So throughout cancer progression, what we see when you inhibit the proliferation of these activated fibroblasts, what we saw was that the pathology of the tumor actually worsened, and that was associated with the immunosuppressive signature. Here's the data specifically about the immunosuppressive signature. We identified that one through RNA sequencing of the bulk tumors and pulled out a gene expression signature, as well as looking for the proportions of what is considered generally pro-tumor or Treg or anti-tumor T effector cells, and then also the relative histopathology. What's interesting is even though by ablating smooth muscle alpha actin, making the overall tumor worse and creating immunosuppressive signature, that immunosuppressive signature actually was associated with with CTLA-4 upregulation. So thereby treating the mice that have then been ablated, depleted for alpha-SMA positive cells with anti-CTLA-4, not only were we able to rescue the immunosuppressive signature to where you now see that the depleted cells now associate and rescue the immunophenotype, but that is actually associated with an improved overall survival and corresponding histopathology. So again, suggesting that modulating the immune system under specific context may actually be beneficial towards the end. Further investigation from colleagues of mine, also at my postdoc, Yang Chen, he specifically looked at the role of collagen from these alpha-SMA positive cells. And in many cases, he was able to phenocopy the phenotype of a worsened pathology and poor survival. And Yang at this time did single cell RNA sequencing in addition to a lot of beautiful immunohistochemistry and flow cytometry, and dug into this, that there was a flip between a loss of T cell populations and an increase in suppressive myeloid cells. And so again, showing it's not just the alpha-SMA, but it's also the collagen specifically coming from the alpha-SMA cells that are involved in recruiting or associated the different immune cell infiltration. And the idea is that it may not necessarily be the collagen itself, but instead what could be decorating the collagen, that integrins or specific cytokine pathways that are associated with that. But it's suggesting again that these alpha-SMA positive cells, one of their primary functions in pancreatic cancer progression is to restrain the tumor, not promote it, and or not form a barrier that stops it, that the collagen itself is actually important in restraining the tumor. Well, alpha-SMA positive fibroblasts are not the only cells within the fibroblastic population of the microenvironment. There's another population that our lab focused on, looking at FAP positive cells. This work was also contributed heavily by Josephine Darploul in collaboration with Kate and McAndrews and also Yang again. And what this group found was that, again, the fibroblast positive cells was a distinct population, as you can see here, quantifying the mice and also in human quantifications, that there's very few co-positive cells on the protein level. However, what's interesting is that when the FAP positive fibroblasts were depleted using a very similar thymidine kinase model as the alpha-SMA cells, that caused a prolongation of survival. The mice without the FAP positive fibroblasts did better. Trying to see the differences between these two mouse models, one of the things that was identified using CyberSort of the single cell RNA sequencing and the GSEA analysis, it looked like there was an opposite effect of FAP on the FAP depletion on the immune system than what we saw with alpha-SMA, suggesting again that all roads seem to be leading to the regulation or interaction of these fibroblasts in promoting pro or anti-tumor environment in the progression of pancreatic cancer. So all of that work's been published. The references are listed in the slide if you want to know more details. But to summarize at least the fibroblast heterogeneity, what we can see in the tumor microenvironment, that not all fibroblasts perform the same function, that the activated alpha-SMA positive fibroblasts and the collagen they secrete are promoting an anti-tumor immunity, whereas the FAP positive fibroblasts seem to be promoting tumor progression. This suggests to borrow the term, a surgical approach, that targeting the entire microenvironment with a sledgehammer may not be appropriate, but instead specifically targeting a subtype of fibroblast, in this case FAP, would be a better strategy, and leaving those alpha-SMA positive cells there to help restrain the tumor. Another thing that was really identified through the study is that pancreatic cancer is often cited as being immunologically cold, that especially when you compare this to other cancers such as melanoma that respond readily to immune checkpoint blockade therapies, the pancreatic cancer has far fewer T cells around. Our studies, by modulating a fibroblast population and seeing that all of our phenotypes continually pointed to the role of the immune system, suggests that the T cells are still serving a function in the pancreatic cancer microenvironment. I wanted to ask the question in a patient-relevant cohort, does this T cell infiltration and maybe the subpopulations of those T cells, how does that affect survival? And can these T cells actually get in with functionally relevant distances? So this is where my journey in starting to study the spatial biology of the tumor started to take off. So what we did, we had access to 132 pancreatic cancer patients who were treatment naive at time of curative intent surgical resection. The patients then went on to receive the standard of care chemotherapy with or without radiation. And then sadly, because it's pancreatic cancer, all of the patients, we had the report of their survival. Those patients' resected tumors were then put into a tumor microarray. We had 2 tumor cores per patient, as well as non-involved pancreatic tissue that was also resected at the time. We then went on to use an 8-color multiplex immunohistochemistry and further analysis, again, asking that question, what about T cell infiltration and how does that impact survival? To quickly go over the panel that we used cytokeratin 8 as an epithelial marker to mark the epithelial cancer cells. We again looked at alpha-systein collagen based off of our previous work, as well as what's nowadays considered a very small T cell panel, looking at general CD3 cells, CD8 positive cells, CD4, as well as FOXP3. Combining all of these markers with cell segmentation, then we were able to identify the specific cell phenotypes. Here we have CD8, CD4, FOXP3 double positive, and also triple negative CD3 positive cells, which with this analysis, we couldn't determine further what they were. The hypothesis is they could be gamma delta T cells. Relatively rare, as well as cancer, we were able to identify normal cells and as well as the general grouping of other, which will include fibroblast, myeloid cells, other things not captured in this panel. Okay, so when you bioinformatically look at things, one of the first things you do is count, right? So that's one of the first things we have. We have the cells segmented and we counted the cells. And what we saw was there was a significant increase in T cell populations in the pancreatic cancer samples compared to their non-involved pancreas. So again, right there, it does show that the immune system is responding to the presence of a tumor in these cancer specimens. Um, One of the things that, forgive me, that I left out of the study, out of the slide presentations, but is in the publication, is that even though the population, the overall numbers of T cells are higher in the pancreatic cancer, what we do see is proportionally a reduction in CD8 positive T cells and an increase in the T helper populations. So suggesting even though the immune system is responding to the presence of the tumor by being a tumor microenvironment, the types of cells that are there are starting to change. Okay, so now to that primary question, what happens to survival? What you can see is that we have significant correlation of survival when we split the patient population on mean for the counts of CD8, CD4 cells, and really just all T cells. Now, the FOXP3 positive population was trending. What we found was that when when you have a presence of all T cells, of any T cells, you have a higher presence of all T cells. And when you specifically look at the ratio of T effector to T reg, which is more associated with is it pro-tumor or anti-tumor immunity going on in the microenvironment, there is no separation of survival at all. Okay, so this is all numbers, just the number of T cells. More T cells of CD4 positive, FOXP3 negative, and CD8 positive cells are associated with improved survival. What about spatial association? So we have investigated this, and this is in collaboration with Dr. Arvind Rao and his graduate students, Dalu Yang and Saptak Bodua. And what we did is that we quantified spatial infiltration in 2 different ways. These are both derivatives of the K function, which comes from ecology, and it is the G function, which can be viewed as a binary. Either you have infiltration or you don't, or the L function, which is more nuanced, where you have low versus high infiltration. We ran this analysis and asked the question within a cell-cell contact radius of about 20 microns. That was selected because your average T cell is about 10 microns in diameter. And so if you're looking at a 20 micron diameter from the center of each of the nuclei, you are having a high chance of catching a membrane cell-cell contact. And so when we looked at that, And then here is an association of what the L function actually looks like. You can see that at the low area under the curve represents low infiltration, medium and high infiltration. Here are the red dots are the T cells associated with the cancer cells. When we look at those associations, what do we see? The only immune cell population that was significantly associated with survival at this narrow radius were CD8 positive T cells. And this biologically makes sense. CD8 positive T cells primarily function through cell-cell contact. You know, they touch the membrane, they have perforin that punches a hole through the membrane, and then they insert glycoenzyme B and they poison the cells and they kill it. Another way that the literature points this out is a kiss and run. They smack it, kill it, and off they go. Whereas the T helper variety, which is the CD4 positive T cells, their primary function is through secreting. This will become important in a little bit more. So this is separate survival. Let's take it another step further. What about multivariate analysis on the clinical level? So multivariate analysis asks the question: are these factors independent of each other, or are we really just catching the same thing? And that, for instance, chemotherapy also is associated with T cell infiltration. And what we found in this cohort, clean surgical margins, whether or not the patients had lymph node positivity and whether or not the patients received adjuvant chemotherapy, were all associated with survival, which is well known in oncology. And we also found that when we were looking at spatial infiltration, only the binary G function was significantly associated with survival still, meaning that the addition of whether or not the patients had CD8 positive T cells close to their cancer cells was an independent prognostic factor of their survival. And to date, this is one of the first and only spatial associations that is actually independently predictive of survival. And this is in a cancer that most people believe to be immunologically cold. So it's very important to look at not only that the cells are there, but where they are. Adding further to that, I mentioned that analysis, as we focused on 20 microns, we actually did the analysis all the way out to 150 microns. And interestingly, the CD8 positive T cells significantly separated or associated with survival in those radii that were immediately close, but not at farther radii. Whereas the CD4 positive T cells also significantly associated with survival, but at farther distances. We're now talking more like the 40 to 80 to 100 distances, which matches with the cell's ability to secrete cytokines and other interleukin factors in order to activate the immune populations. And this kind of leads to like future directions that we're working on is that, all right, well, these are just two-way, right? One cancer cell communicating to one T cell and how they're associated with it. Now to work on multiple relationships and whether or not those T cells are there based off of other microenvironmental conditions. Well, speaking of other microenvironmental conditions, we did investigate that. And what we looked at was, did the presence of alpha-SMA, or therefore it's collagen that is likely secreted by those alpha-SMA positive fibroblasts, did that inhibit the infiltration of T cells? Because a common belief in the pancreatic cancer field is that the collagen forms a wall, a barrier that the T cells can't get through. And that's one of the reasons why pancreatic cancer may be immunologically cold. So to investigate that, what we found was when we were quantifying the intensity of the pixels in that narrow radius, there was no association between the patients who had low T cell infiltration versus the patients who had high T cell infiltration, suggesting that there was no association with collagen or alpha-SMA on that level. To further that, we actually looked at the correlation of how much of that tumor area we were looking at had collagen positivity versus their T cells. So what we found is that we had an increase correlation, positive correlation of the amount of collagen with the amount of T cells, both the CD8 and the CD3, CD4, but they're negative for FOXP3. So now going back to the mice, right? So a lot of this has been in patients, going back to the mice to try to tease that out. We're trying to ask the question, well, what comes first? So in this mouse model, what we did is that we had a pancreatic cancer mouse model, and then we depleted both the CD4 and the CD8 positive T cells through an antibody that depletes the cells. The top panels are your flow cytometry that shows that, yes, we successfully depleted the CD4 and positive T8 cells. And what we found that it didn't impact whether we had alpha systeine collagen there at all. However, when we did the inverse, excuse me, so we already know when we did the inverse, when we ablate alpha systeine collagen, we see a reduction in these positive T cells, suggesting that the cancer fibroblast happen first and that they are then recruiting the T cells in. But what happens if we just remove the T cells? What happens to the phenotype? What we find is that we then phenocopy the alpha systeine depletion phenotype, where we have poor histopathology, and it's also associated with higher invasion. So in summary of this portion, focusing specifically on T cells, we asked the population the question, does T cell populations affect survival? The answer to this was yes, that high T cells in counts correlate with survival, and that high CD8 positive T cells, and later analysis showed CD4 positive T cells, their infiltration in areas around cancer cells did independently predict survival. And at this time, that the T cell infiltration was not inhibited by alpha systeine or collagen's presence. So the next question is, well, what about the cancer cells themselves, right? All of this time, we've not changed the cancer models that we were working with. They're all KRAS and P53 driven. What is it that is going on in the cancer? Are the cancer cell subpopulations interacting with the microenvironment? And then how does that impact metastasis? Well, when you start looking and you thinking about cancer cell subpopulations and metastasis and chemotherapy and immune microenvironment, as a cancer biologist, one of the first things that you think about is the epithelial to mesenchymal transition, EMT. EMT in the cancer biology field has been associated with chemotherapeutic resistance, migration, metastasis, and immune evasion. EMT has also been identified in pancreatic cancer RNA expression profiles, where there's been lots of studies trying to profile pancreatic cancer, similar to the way that we can profile breast cancer, and then associate the different therapies that are best benefited for those patients. In general, across most of these studies, there's a consensus of two. There's a classical phenotype that comprises a good 3/4 of the patients, and then there is a second subtype that goes by multiple names: basal, quasi-mesenchymal, squamous. So you can tell from all of those, they're associated with an acquisition of mesenchymal features. Okay, so how do we study this? Well, EMT, though often referred to as just a single entity, is a very complex and context-dependent path mechanism. Many different transcriptional factors can drive it. Some are important in some cases, in some cancers, and some are not important in the other. Also, EMT is not a binary thing. It's not epithelial or mesenchymal, but it is really a spectrum of lots of different transitional states. And in general, cancer biologies view this partial EMT, this intermediary step, as being the most relevant. That's where the cancer stem cell hypothesis is associated with that partial EMT step. Those are the ones that are less proliferative associated versus full mesenchymal transition. And I pause to explain this, is that where the studies focus on this spectrum, whether they're looking at partial or full mesenchymal transition, you're going to come up with slightly different answers looking at EMT. And so it's a very complex environment. So for the future studies that I'm talking about, I want to highlight three of the most common transcription factors that are well studied. It's SNAIL, that's two different proteins there, SNAIL and SLUG, as well as TWIST1 and 2, and there's also ZEB1 and ZEB2. All of these are studied quite a bit in cancer biology. So one of the first things that we wanted to ask is, okay, again, looking at that spectrum, if we're not looking at the overt mesenchymal or sarcomatoid tumors, is EMT still being able to detect it when everything else about the tumor morphology really matches the epithelial morphology? Can we actually detect EMT? And for this, we mined a single cell data set. We isolated out the cancer cells, and we did magic on this from isolated from, this is from Donna Pierre's lab, where they fill in the holes that can happen in single cell RNA sequencing. And in this, we fed it a 315 universal EMT gene signature. This gene signature was published by Jean-Paul Thierry's lab in 2014, where they looked at the microarray data of hundreds of cancer cell lines and tumors, and they came up with what was considered a universal EMT signature, and we applied that signature. One of the things that we noted is that when applying this signature, we get a full spectrum of epithelial to mesenchymal cells through this. Another thing that is interesting is that in general, some patients correlate to be more mesenchymal. For instance, you have patients 36 here in the bottom left-hand corner of the image of the UMAP, where's more mesenchymal, which is like, patient 40 is much more epithelial. And then in a bunch of the conglomerations of patients in the middle are more heterogeneous. We double checked that these EMT scores did not correlate with the pathology score in any way, suggesting that these EMT associated phenotypes is not something that we're going to pick up in standard histopathological score. This particular data set that we mined did not have survival analysis. So we wanted to then ask the question, okay, from first principles, what does this EMT score do in a patient cohort that we have survival analysis? In order to address that question, we went to the TCGA. It's important to note that the TCGA is, again, resected tumors, much like the single cell data set, not representing metastatic tumors. Again, we saw no correlation with this EMT score associated with their pathology score. But surprising to us, what we found is that it did significantly stratify survival. And what was even more surprising was that it stratified survival opposite to the way that you would think. Patients who had a mesenchymal status here represented by low TAN score or the red line had improved survival. And this is important to note that in the TCGA, those other gene signatures that I mentioned of classical basal, none of those signatures separate survival in the TCGA cohort. Well, like I mentioned, TCGA does not have metastatic tumors. What if what we're seeing is really a metastatic phenotype? And let's look at this. So then we mined another data set. This one's bulk RNA sequencing from a rapid autopsy program where you have matched metastasis to the primary. And when we're looking at this, what we see that the primary tumor has a wide spectrum of EMT signatures, but as you go to distant metastases, more and more of those metastases are associated with epithelial phenotypes. The peritoneum and pancreatic, so local kind of metastases are more mesenchymal, whereas the lung and the liver, especially the liver was the most significant, was more epithelial. Now, this is interesting because clinically, it's well appreciated that patients who have liver-only metastasis versus lung metastasis do considerably worse. So again, that the liver metastasis is a poor prognostic factor. So we then wanted to ask the question, well, what is EMT doing, perhaps with organotropism? So this is kind of a summary of Nature first author paper, as well as a paper that was published in Cell Reports, and as well as another one in preparation. I'm just going to save you a whole lot of bar charts and summary points to kind of describe the models that we did here. So we have two different versions of the KPC mouse model, which is the standard use model for studying pancreatic cancer. It has the driving mutation of KRAS G12D and an amplifying mutation. This one is a dominant negative mutation in P53. It's a very metastatic mouse model. If you instead remove P53, you have a relatively low metastatic mouse model because the primary tumor is very aggressive, but you rarely see metastases. And then we took those two base mouse models and we did a series of cancer cell specific knockouts. And the design of the study is to be a loss of function of EMT by knocking out the EMT driving transcription factor SNAIL and TWIST, or another EMT driving transcription factor ZEB1, or a gain of the EMT function by knocking out the adherent junction E-cadherin. The loss of E-cadherin is considered a hallmark of EMT. So we forced EMT by removing E-cadherin. to again shave you all those bar charts and quantifying the EMT, this schematic shows you where each of those models show up on that EMT spectrum. The baseline KPC mouse models have some level of EMT, but they are generally epithelial. When we remove the EMT driving transcription factors, we stabilize an epithelial phenotype. When we remove EK-adherin, which is an epithelial marker, what we do is we start pushing more to a mesenchymal phenotype, and we can push that all the way through a full loss of EK-adherin. Okay, so now we have all these models that have various controls and stabilizations or induction of EMT. What does that do to metastasis? So again, to summarize this, what we see is when we stabilize epithelial phenotype, we're seeing an increase in liver metastasis. That increase in liver metastasis was primarily not by incidence, so the number of highly metastatic mice who get it, but more by the area when we're in the KPC model. When we look at this in the low metastatic mouse line, we also see an increase in the incidence of liver metastases. Whereas when we push mesenchymal characteristics, we see an increase in lung metastases and a decrease in liver metastases. And when we push it all the way through an EK-adherent full knockout, what we see is a heightened increase in the incidence. So all of the mice had microscopic lung lesions, but only the ones that maintained their plasticity, so they didn't have EMT fully knocked out, created clinically relevant metastatic lesions. So the metastatic lesions grew out and continued the colonization. So in some ways, this really supports the EMT dogma that, that the cancer cells undergo EMT transition, then they localize primarily to the lung, and then they switch back. They go to the inverse to MET and take on epithelial characteristics or able to grow out. If you remove that ability, the cancer cells have an inefficient grow out in the lung. However, in the liver setting, that doesn't seem to be the case. And interestingly, in collaboration again with my colleague Yang Chen, Yang used a dual recombinase mouse model, which allows us to drive cancer by the flippase and also to label or modify other cell populations. Yang crossed the dual mice with also a dual reporter, so you can get all of your cancer cells, which are flippase or green, and all of your other cells, in this case, would be alpha-SMA or FSP1, which are fibroblast or mesenchymal markers, will be labeled red. So if these cells had to undergo alpha-SMA or FSP1 associated mesenchymal transition in order to metastasize, the metastatic lesions would be permanently red. That's the model. So what does he see? Well, when Yang looked at liver metastasis, all of the liver metastases were green. Not a one of them was red, suggesting that those large liver metastatic regions did not turn on alpha-SMA FSP1. He was able to identify red cells in the liver, but they were all in the micrometastatic setting. So suggests that the liver microenvironment, specifically in this mouse model, epithelial to mesenchymal transition was not necessary, at least for the alpha-SMA and the FSP1 portion. Okay, so identifying this further, let's go into this more. The KPC and snail twist mouse models, we did do single cell RNA sequencing. We analyzed this in the same way that of the patient data that I presented earlier. We isolated cancer cells, ran that same 315 gene signature. And what we found was that across these mouse models, we were able to recapture a full spectrum of EMT cancer cells, similar to what we saw in humans. This also did not correlate with the overt histological morphology. So in general, the KPC mice and the KPC mice that had Snell and Twist knocked out, their overall histopathology score was mostly epithelial. So now, because we have the single cell RNA data set, we also have a data set where we isolated out the YFP expressing cancer cells and enriched for them, and also did microarray. These two data sets combined, we're able to start looking at RNA expression profiles, looking at the differences between EMT stabilization and not. To first focus on the primary tumor phenotype we identified, what we found was this association with immune cell regulation, whereas the cancers that are epithelial stabilized, they have different mechanisms of immune cell association than the cells that are more EMT. I also want to throw this one point out that by looking at the bulk gene expression from the sorted out cancer cells, we were able to detect more gene signatures than we were in the single cell. And those gene signatures were specifically associated with the metabolic processes that may be more subtle to catch. So I know a lot of people are really focused on single cell RNA sequencing. There's certainly a lot of benefit to it, but sometimes the reduced amount of RNA that you get by the necessity of looking at a single cell, you are potentially missing some important biology that could be investigated because it's a lower frequency signal. So just kind of putting that out there, that looking at the heterogeneity of needs multiple ways of investigating, not just single cell. It needs to be able to investigate it further. So off the soapbox for that moment. Now moving on, what had happened? So let's now count the immune cells that were isolated in the single cell. On the human side, when we group the patients to more mesenchymal, kind of a partial phenotype or epithelial phenotype, there is maybe a trend down into the mesenchymal phenotype, not necessarily something that we would normally follow up on the mouse single cell data set because it's cleaner with the genetically engineered mouse models. What we found was a very big difference in the number of immune cells, where our KPC tumors, so the control tumors that had EMT fully capable versus the Snell twist knockout, so it's epithelial stabilized, at the early time points, they were very similar in their immune profiling. For instance, if we focus on the T cell where the black arrow is, they both have comparable amounts of T cells in their microenvironment. But as the tumors progress to late or aggressive tumor varieties, the KPC late one that has a very mesenchymal phenotype, the mice that we sequenced, they almost completely lost their T cells, whereas the snail twist tumors maintain their T cell numbers. So this is with RNA sequencing. This is out the context of spatial. This is, of course, a spatial lecture. So we then go in and start looking at the pictures. We go back and mine that human data set I mentioned, as well as creating a corresponding mouse staining signature for mouse FFPE. And when we quantify the EMT in these patients, because remember, we have a mesenchymal marker in that panel of alpha SMA. So we're looking at the cancer epithelial cells that are co-positive for alpha SMA. So we're quantifying partial EMT. What we found was an we see a similar pattern, again, only in the KPC mice, because the snail twist mice, we've, of course, stabilized the epithelial phenotype, and we don't really have alpha SMA positive, co-positive cells there. So digging further, let's now look at the spatial association. And what we found is that the partial EMT cancer cells in the patient cohort had significantly fewer T cells around them than the ones that were full epithelial, suggesting that one of the mechanisms that mesenchymal or partial mesenchymal expression T cells, cancer cells, is to inhibit the infiltration or perhaps the recognition of the T cells into it. Whereas the epithelial phenotype, they're still progressing. They're still evading the immune system. They are employing different mechanisms of immune cell invasion. Well, this is in the primary tumor. What about the metastases? So what you can see from this summary plot of what's up and down in the GACA analysis, when we're looking at the metastatic cells versus the primary cancer cells, we can see that, again, the metastatic cancer cells have different ways of interacting with the immune microenvironment and with metabolism, and they're also more migratory in general. And so to investigate, some of these further, what we did is that we actually do have some single cell RNA sequencing data from these matched liver metastases. And this is continuing work that my graduate student Ayushi Mandaloy is working on. And when we're looking at the liver metastases, we can see, focusing on the T cells, yes, we do see that big difference, no T cells versus a lot of T cells in the primary, but they're matched metastases. The signature is about the same. The effect of EMT in the metastatic setting does not seem to have an effect on the metastatic microenvironment. Well, this is again RNA. What about the protein? Are we seeing something similar? So here is many more multiplex immunofluorescent staining panels. So each one of these are about an 8-plex. And what this is that you can see matched primary with the liver metastases. The top panel is quantifying EMT. You can see the farthest left panel is that we have one lesion here that has very few EMT co-positive cells. And you can see in that lower panel that that is associated with more CD8 positive T cells in that microenvironment, as indicated by the white arrow. And then there are other areas of the same primary tumor that are very EMT. We have a lot of red and co-expression of the epithelial markers with the mesenchymal markers, and that has very few immune cells, if any, in that region. And then the matched metastases, you can again see that the metastases in the liver are very epithelial. However, the immune cell components of that epithelial cancer are different. And here, indicated by the white arrow of the liver section, the immune cells seem to be pushed off to the side. They are CD3 positive and CD4 positive, but almost no CD8s. Digging into exactly what these CD4 helper populations are, what's their spatial association, as well as how they secrete cytokines is what the dissertation focus of another graduate student in my lab, Christina Larson. She's just getting started, and this work is going quite well. So this is all on the mouse side. What about the human patient side? So one of the things that's not well appreciated if you're not in the field, because most of the patients walk in the door with metastatic lesions, very few of those patients have their metastatic lesions removed or even their primary tumor removed when they're already metastatic. Most of the patients just get honestly palliative chemotherapy to try to extend life, but it's not a view to be curative. In collaboration with Dr. Ashik Masood as well as Dr. Basil Reyes, they have access to a unique patient cohort where they have resected primary tumors and matched metastases. Ashik and his group then took these matched sets, also with lymph nodes and normal adjacent pancreas, and they did Visium spatial transcriptomics on those patient sets. And from there, they were looking at their cell type markers, and they deconvoluted and created what they call were spatial ecotypes associated with it. This paper was recently published in Nature Genetics, and please go read it. It's much more detailed than bioinformatically than I'll get into here on the biology. But on the biology side, there are a couple of key points we would like to highlight. One, the spatial ecotypes of the primary tumor versus the metastatic liver tumors were fundamentally different cancers. Even though genetically the malignant cancer cells themselves were very similar, the microenvironment was very, very different. Whereas the primary tumor had an increase in fibroblast and immune signatures, and they had an EMT signatures and immunosuppressive microenvironment, the liver had a higher amount of cancer cells, lower amount of stroma, and they also had this metabolism phenotype as well as just a lack of a lot of immune cells. Interestingly, though, matching again to what the mouse data is that I just showed, is that there was a tumor liver invasive border that was filled with immune cells. And these immune cells were a split of pro-tumor and anti-tumor immunity. And in the paper, we discussed that this seems to be a tug of war of a push and pull at this invasive barrier. And there could be a spatial barrier, a set aside, that then stops those anti-tumor immune cells from infiltrating into the liver metastatic tumor nodule. So further investigations into this using these mouse models that now seem to recapitulate what we've identified in the patient setting is a current focus of both of our labs. So looking at this again, what are some of the ways, what are some of the tools in the last few minutes of this lecture that we are implementing? So we're really interested in this metabolic phenotype that I've mentioned that could be associated, but how do we study metabolism in space? Well, there's a couple of ways that myself and my collaborators are investigating this. This is, again, a work of a third graduate student in my lab, Meet Patel, and his co-mentor, Dr. Anna Sarasi. And what Meet and Anna are working on is doing dual metabolic PET imaging. So we are imaging with the standard FDG glucose for capturing glucose metabolism, as well as looking at C11 acetate. And acetate is shuttled into the Krebs cycle. And so by having an increased amount of acetate uptake as well as processing, that can be a surrogate for higher levels of the Krebs cycle function or oxidative phosphorylation in addition to just glucose uptake. And the idea is that by computing the ratio, we'll be able to see are the cancer cells or the tumor region undergoing just high glycolysis, or is it also going all the way through the oxidative phosphorylation? And one of the things that is unique to what we're doing is that we have all of these different cell lines isolated from these EMT mouse models, and these cell lines have a very distinctive epithelial or squamous features. And what we're doing is that we're not only just looking at the ratio, but we're also looking at the subtumor localization and seeing if that correlates with blood vessels or the type of histomorphology, and aligning these tissues in space and correlating them with their PET imaging. As well as we will be then doing multiplex immunofluorescence and looking at the immune infiltration, the blood vessels, the stroma on that. So stay tuned. The experiments have been run and we're in the data analysis side. So we're looking forward to getting this data published within the year. Following up on that, looking at more the metabolic, the OXPHOS, as well as lipid metabolism, we're also doing mass spec imaging. And this one was particularly a DESI imaging, to where we have optimized the ability to do multiplex immunofluorescence on a snap frozen tissue in order to capture more of the lipid moieties. And that analysis is continuing. But what we can see is that we get some of the masses that are specific to some types and some of the others. And then we'll be able to associate those masses specifically with immune cells, stroma, or also the cancer cells, and tease those out. And then a final point is that we have all of these various different EMT stabilized cell lines. They also metastasize spontaneously to specific spots in the mouse immune system. And there is a correlation with where those cells metastasize to, as well as the type of metabolism they go through. As you can see through the top heat map, there's a very big difference in the types of lipids associated with these cancers. So this is something that we're actively working on. Again, looking at metabolism and lipid omics in space, and how does that correlate with the EMT spectrum and metastases. So to quickly summarize, to quickly go through one more piece of work that we're going through is, what about chemotherapy resistance? Here we're looking at a novel pan-RAS inhibitor. When the mice, the slide disappeared, but what we see is that when we treat the mice with a novel pan-RAS inhibitor, we have a significant decrease of the tumor volume, and that decrease is also associated with an increase in immune infiltration. However, that immune infiltration is specific to the cell line being used. So it's not that the KRAS is recruiting more immune cells, it seems to just be controlling the cancer cells. So this cell line is a high immune infiltration in cell line. When we reduce the amount of cancer cells through the KRAS inhibition, we have a higher proportion of the immune cells. However, this tumor that has very low immune infiltration in general, we don't really change the proportions. What about spatially? When we treat the cancer cells, what we see is that even though we do not alter EMT status, we are also altering the immune infiltration. And so what we're seeing on that front is the spatial differences is that, epithelial cancer cells, so the dark green ones, have more immune cells in that radius. Is significantly different between the groups. You see these same radii plots that we plotted before. We're also seeing a significant difference in the immune cells around specific fibroblast. So in this case, the blue fibroblasts are those that are associated with alpha-SMA and Desmond positivity. They're still associated with more immune cells infiltrating around them, whereas the CAFs that are FAP or are integrin beta-3 positive, they are not associated with any sort of immune infiltration, and we're quantifying that. For all of my bioinformaticians out there, this is the way that we quantify that data, and looking at the infiltration associated with various different positivity. I'm happy to go into this more during the question period if you'd like to see how we're developing those analyses. So in summary, spatial relationships between cancer cells, the T cells, as well as the fibroblast, are a determining factor in pancreatic progression. Fibroblast can aid immunity regulation. Cytotoxic T cells within proximity of cancer cells is independently predictive of survival. Partial EMT is one mechanism for fewer T cells in PDAC tumors, and cancer cell inhibition can be enhanced by already present tumor immunity localization. And this opens the door for TME targeted therapy combinations, either through checkpoint blockade or through mechanisms to specifically target integrin beta-3 or FAP. And with that, I'd like to thank my collaborators, the funding that enables the work, as well as my amazing lab. The people who've been doing the most recent work were highlighted. And if anyone would like to contact me, you can reach me on Twitter as well as my email. And with that, I'd love to take any questions.
Julie, this is like that is a lot of work. That is like amazing. That is a tour de force in looking at this. You should be very proud. You and your team should be very proud of the work that you've been doing. This is really quite incredible. It looks to me like you have got data coming out of your ears. And I'm curious about your interactions with any colleagues. You mentioned a few bioinformatics colleagues, and I presume that you're also interacting with folks who are interested in data science and applying kind of predictive models to some of these data types. I'm curious if you might relay a little bit of your experiences about things you've discussed with those folks or ideas and directions you might be going in.
Yes. So as you said, I've got more data than I can handle. By at least bioinformatically, we can handle. And there are always active projects that so for instance, I think I collaborate with about five different bioinformatic or data science colleagues are each we're investigating, we're taking these data sets and designing new methods or models in order to investigate it. So for instance, looking at time and space, looking at 3D spatial infiltration, because a lot of the analysis that I discussed was focused on 2D space. And so, well, what happens in 3D space? And then even in 2D, well, I don't just identify T cells. I'm also a co-director of our spatial biology core, where we have the Comet as well as Visium and Xenium, and one of the many hats that we wear is like, my lab, we're developing 50-plex markers to fully phenotype the mouse immune system, similar to how we do in flow cytometry, but now in space. And that's, associated with a lot of Christina's work. So now we not only know that the cancer cells are there, where they are, and the T cells. Now I know if there's a macrophage there or not, or a fibroblast, or how close you are to the immune system, to the blood vessels or the lymphatics. All of these are being quantitated, but how to mathematically pull out meaningful information is something that it's wide open projects. If people are interested, if they've got ideas, I've got data. We're very collaborative. We can share the data, we can work on it. And I'm very interested in the biological relevancy. And of course, if you come up with something that's important, kind of like those plots on, we were just looking at KRAS inhibition, and then we found all of these associations that the literature has known for a long time. We didn't expect to see that. Well, that now means, well, KRAS didn't cure the mice. Okay. The mice still had tumors. It slowed it down. Can we cure the mice if we now add checkpoint blockade? Can we cure the mice if we now ablate those fibroblasts that may be inhibiting the immune cell infiltration? So those types of questions, we now as biologists, as someone who's a mouse modeler and has access to a lot of those models, we can now follow up. And so that's what we love about bioinformatically pulling this stuff out, because I have a lot of ideas, but mathematically, I'm not an engineer. I don't know how to make the data do that. So this is a fantastic collaboration. We have, like I said, I have a lot of data. Anybody who's interested in mapping or anything like that, there's a lot. There's a lot to work on. And normally my collaborators, they go, ooh, I'll take one of these questions, and they work on it, and it becomes often a dissertation project or one of the projects for their students.
I think one of the things you just mentioned in your answer there was those GNL functions which you have leveraged. Those are, I presume those are in 2D. And I was, as you were talking about them and describing them, I was kind of wondering, oh, you've got that radial distance, that threshold within which you find the cells in relative to the cancer cell, that is actually a three-dimensional distance. It looks like you're probably maybe miscounting when things get a little too close, because they could be actually behind it. They're just projecting through. I mean, correct me if I'm wrong, but that may be an opportunity to look at things in that third dimension to improve your cell counts. At the same time, you've also got that temporal dimension. I mean, all of this metastasis takes time, and that process evolves over time. If you could capture that process, that gives you an additional information about, say, the rate of cell density that subpopulation and that microenvironment, attracting those cells over time. That's an important value, I would imagine.
Absolutely. And so some of the ways that we are doing this, and actually my students and I, the aspect of time and the timing of the immune infiltration is quite important. There are multiple ways of quantifying time. You can group them into longitudinal sampling. You can also group them into longitudinal labeling. And I kind of like the idea of longitudinal labeling
Mm,
because then you can label them over time, but still analyze a snapshot. Versus, and you can combine those, you can label them over time and then collect over time. But the negative about that is you're never actually studying the same cell and seeing how it's progressed over time. And so having to combining those, there's techniques already to do these, but adding the time and space together. So that's one of the things that we're quite interested in. Our mouse models, we have lineage tracing. We can inject tracers also into the mice and label them progressively over time. A lot of the genetically engineered models, they can be drug inducible to induce the labeling. And so we can track these over time, and we're very interested in that. Um, Analytically speaking, though, there's a problem of multiple assessment of the same cell, right?
yeah.
How do you pull out statistically the really meaningful thing, right? How do we know the effect of that immune cell at this particular point in time versus a summarized point of time over time? So that's mathematically, I don't know how to address that. Again, is wide open for the mathematical questions. To go back to the 3D point, a lot of this is limited on the instrument that you collect the data on.
Mhm, yeah.
So a lot of this is that I showed that the spatial is really a single plane of a 5 micron tissue section. So yes, it's 5 microns. There is 3D associated with that, but Z stacking, where you would
Mhm.
image all the way across it, is not normally done. So instead, what is done is that you take a single focal plane and then everything's viewed as a focal plane. So it's not that we can't do Z stacking. It's not that we can't go in deeper. With the PET imaging, we definitely, take as many slices as we possibly can, but the PET imaging is lower resolution than a microscope. So it's a lot of those is starting the collaboration up front. It's not waiting until I've collected this amazing data set and then I go, hey, answer my questions. Whereas if we had started that bioinformatic conversation up front, I could have collected my data in a different way. I could have said, all right, it's worth creating a huge amount of data to do this as a Z stack.
Do-
Or let's do this as a Z stack where we have the 3D all the way through the 5 microns, but use it at a lower plex. So it's not so much data that we can't understand. So having those conversations, collaborating with people early is really important. And one of the things that I've learned that as a biologist, I generate data slowly. It takes a while to recruit patients, to generate the data sets, to grow the mice. Where as my bioinformatics colleagues, they're like, well, I'm ready to go. And so that's one of the things that my collaborations with Arvind and my collaborations with Satwik and Dean here, so 3 people in particular, we talk often over a long period of time. And I pitch things to them when I just have an idea. I've not done any experiments, and getting their input at that stage has been amazing. Because there's so much things I could do to provide them the data that they need to do amazing things in their field, but I have to know about it up front. And so I would recommend that people start those collaborations early, and it's a cultivated collaboration. The bioinformaticians, the data scientists, they're not just there to answer your questions. They have questions of their own. They have new techniques that they can create, but you got to talk to everybody early.
I think that's such an important point. Thank you for bringing that up. Is that this is a team science enterprise, and that people with your skill set clearly, you can look at these questions, you can collect these data in the ways that you're collecting them. Had you kind of included the data scientists, the bioinformaticians, the quantitative folks from the get-go, they may have had some suggestions that may have helped you improve your data collection methodologies to help streamline some of their analyses and make them more, allow you to make greater inference on them. So thank you for bringing that up. Julie, I feel like I could ask you a million questions all day long because you've got so much stuff there. And actually, I took a whole bunch of notes, but I just don't have time to ask them. But I want to thank you so much for sharing your amazing amount of work with us. You have an enormous amount of data. I would hope that anybody listening who has big ideas about it would reach out to you. But in the meantime, thank you so much, Julie. We really appreciate you sharing all that. Have a wonderful Friday and a wonderful weekend.
All right. Thank you so much. And again, if anybody needs to contact me, on the slides are my Twitter handle and my email, and all of the data sets publicly available. But if you want insight that's, not the raw data, please feel free to contact me.
Thank you, Julie.
Thank you. Bye-bye.